Imagine a world where we can edit our DNA like a text document, where genetic diseases can be cut out and healthy genes pasted in. Sounds like science fiction, right? Well, with CRISPR technology, this futuristic concept is quickly becoming a reality. But how does this incredible technology actually work? Stay tuned to find out, because what you're about to learn might just blow your mind. Before we dive into the mechanics of CRISPR, let's take a trip back in time to 1987, when a group of Japanese scientists stumbled upon something peculiar in bacteria. They discovered repetitive DNA sequences, now known as CRISPR, but at the time, no one knew what these sequences were for. So, what is CRISPR really, and how did it evolve from a bacterial defense system into the revolutionary technology we know today? The answer to that mystery took almost 2 decades to unravel. But when it did, it changed everything. CRISPR technology isn't just any tool. It's like a pair of molecular scissors designed to cut DNA at precise locations. But how does it know where to cut? Enter the hero of our story, Cas9, a specialized protein that acts like a highly skilled surgeon, guided by an RNA molecule designed to target specific sequences in the DNA. Once it finds its target, Cas9 makes a precise cut, removing a piece of the genetic code. But here's the twist. This isn't the end. It's just the beginning of the editing process. Because after the DNA is cut, the cell's natural repair mechanisms kick in. And this is where the real magic happens. When CRISPR makes its cut, the cell goes into panic mode, rushing to repair the break. But it's in this frantic repair process that we can introduce changes. Scientists can trick the cell into inserting new genes or deleting harmful ones during the repair. It's like editing a typo in a sentence, except this typo could be responsible for a genetic disorder. Imagine being able to delete the gene responsible for a deadly disease or replace it with a healthy one. Sounds like something out of a sci-fi novel, right? But here's the kicker, it's not just fiction anymore. The potential of CRISPR goes beyond just fixing typos in our DNA. We're talking about a future where we could possibly eradicate genetic diseases entirely. But that's not all. CRISPR could also be used to enhance human abilities, create disease-resistant crops, and even revive extinct species. The possibilities seem endless, but with great power comes great responsibility. While the possibilities are exciting, they also raise a lot of questions. What are the ethical implications of editing human DNA? Could this technology lead to designer babies? Or even worse, a genetic arms race? These are questions that scientists, ethicists, and all of us need to consider as we move forward. As we speak, CRISPR is already making waves in the medical world. Clinical trials are underway to treat conditions like sickle cell anemia and certain forms of blindness. In agriculture, CRISPR is being used to create crops that can withstand harsh climates, potentially solving food shortages worldwide. But while the future of CRISPR is bright, it's also uncertain. The technology is advancing faster than ever, but we're still in the early days of understanding its full potential and risks. CRISPR technology has the power to reshape our world, but it's up to us to steer it in the right direction. What do you think? Are we ready for the genetic revolution? Let us know in the comments below. And don't forget to like, subscribe, and hit the bell icon so you won't miss what's coming next in the world of science. Thanks for watching. Stay curious, stay informed.